pQTL using 8,000 protein quantified in all samples and 10,000 genotype. By using the JAMA, and we identified 318 cis pQTL regulating 464 proteins and 87 trans pQTL at FDR 5%. And I highlighted some of these proteins that are involved in the metabolism pathway. And so for example, there's some drug metabolism, amino acid metabolism. And in our human study, we actually found this cis pQTL is highly enriched in exonic variants. However, in our rat pQTL, we didn't observe the same enrichment. One possibility is that maybe we didn't include all the exonic variants for the mapping. And the next question we say whether there is any co-localization between cis pQTL and cis eQTL. So we performed the eQTL mapping using transcription data, and which was provided by Laura, and we identified 1,200 cis eQTL, regulated around 3,000 transcripts, and then we co-localized this cis pQTL and eQTL. We identified 115 co-localized paired, and many of those show the same leading SNP. And as you can see, the genetic effect shows highly consistent between pQTLs and eQTLs. And so then we try to explore whether any of the cis pQTL regulate protein expression through the co-localized trans QTL. So we perform a co-localization between cis pQTL and the trans QTL. And then we actually identify 45 co-localized cis and trans QTLs. And here is one of the example. And this QTL regulate regulate the expression of the Wang as a trans pQTL. And in this region, we also identify this QTL, regulate the expression of the LRSM1 as the cis pQTL. So although there's no functionally direct connect between these two proteins, but we believe this LRSM1 could be a good regulator of the expression of the Wang. And so lastly, we wanted to link the pQTL to any of the phenotypic variation. So we download all the 234 phenotypes from the network and map all this phenotypic QTL. We identify 68 phenotypes show significant QTLs. And then we co-localize our pQTL with the phenotypic QTL. And we found two of those have a very nice shared QTL. Here is one of the example. And this QTL regulates the expression of the NIP-PLD. And also this QTL associated with the glucose uptake. And NIP-PLD is a enzyme which involved in the lipid metabolism. And we thought this might be a good control of the NAP with the glucose. And then this protein expression actually highly correlated with phenotype values. And so with that, I would like to thank you, all my lab members, and especially I would like to thank you, Laura and Laura, for providing the support for this pilot project, and all the collaborators who contributed to this project. with that, thank you for your attention and happy to take any question.
You want to take that? Can I answer questions?
Xusheng, you have a question?
Yes.
I guess I was going to ask about what are the things that control power here, right? Like I'm sure that there are more PQTLs than what you discovered, and you could generate more sequence per individual, or you could expand to more strains, or maybe there's another access. What are your thoughts?
Yeah,
Satisfied with where you are?
definitely the power is pretty lower because there's only 30 eyes gene from this rat, eyes genes. And if we use larger family like a BXD or CC mice or DO mice, I think the power will be significantly increased. And so we definitely will detect more PQTL if we use a larger family.
Go ahead. I'll shout. I might have missed it. I have really awesome talk. It was really cool. You integrated the EQTL and PQTL that had the same variant, but I might have missed it. What fraction, if you look in the EQTL data set, what fraction of the QTL had a PQTL and vice versa? So if you take the set of PQTL, for instance, how many of them also had EQTL in the same gene or the same variant?
So here we start with all this PQTL and looking for the 1
Recording
megabase
in progress.
1 megabase franking sequence to see if they are shared, or we can say is the same gene or proteins. And then we look at whether this has a co-localized PQTL. And after that, we say if they are co-localized, then whether they share the same leading SNP. And so there's a two level. The first level is whether they show the same pattern of the QTLs, and then we say whether they have a same leading SNP. That's what our analysis.
So the next talk,